Hello, my dear students. Have you ever thought about how your body turns that partially digested food from the stomach into absorbable nutrients? The real magic of digestion happens in the small intestine, but it does not work alone. In this session, we will dive into the crucial roles played by the pancreas, liver, and gallbladder, the unsung heroes of chemical digestion. Together, I will explore how the pancreas releases enzymes to break down proteins, fats, and carbohydrates, the liver's role in producing bile and detoxifying substances, how the gallbladder stores and secretes bile into the duodenum, and most importantly, how the small intestine becomes the site of maximum digestion and nutrient absorption. Get ready to follow your food's journey into the most efficient and intelligent part of the digestive tract. Pancreas, its basic information. The pancreas is a retroperitoneal gland located behind the stomach. It is about 12 to 15 centimeter long and approximately 2.5 centimeter thick. It lies posterior to the greater curvature of the stomach. You can find the location of the pancreas in the adjacent image. Structure of the pancreas is pancreas can be divided into three parts: head, body, and tail. Head, it is a broad and nestled in the curve of duodenum. Body, it is the central part of the pancreas gland, and tail, it is the narrow and tapering end of the left side. Secretion pathways. Exocrine cells secrete pancreatic juice into small ducts. These small ducts merge to form two main ducts. One is pancreatic duct, duct of Wirsung, or main duct, and accessory duct is called as duct of Santorini, or it can be called as secondary duct. Pancreatic duct or duct of Wirsung, it joins the common bile duct from liver and gallbladder. Together they form the hepatopancreatic ampulla or ampulla of Vater. This ampulla opens into the duodenum at the major duodenal papilla. It is located about 10 cm below the pyloric sphincter. Sphincter of Oddi. A smooth muscle called the sphincter of the hepatopancreatic ampulla or sphincter of Oddi. It surrounds the ampulla. It controls the flow of pancreatic juice and bile into the small intestine. Accessory duct or duct of Santorini. It basically opens directly into the duodenum. It is about 2.5 centimeter above the ampulla of Vater. It provides an additional pathway for the pancreatic juice. Pancreatic juice composition and function or daily secretion. Pancreas produces 1200 to 1500 ml of pancreatic juice per day. It is clear, colorless fluid. Composition of pancreatic juice. It has water, some salts, sodium bicarbonate, digestive enzymes for carbohydrate, protein, fats, and nucleic acids. What is the function of sodium bicarbonate or alkaline pH, which is maintained 7.1 to 8.2? It neutralizes acidic chyme from stomach. It stops action of pepsin, which works alone in acidic pH. It creates suitable pH for pancreatic enzymes to act in the small intestine. Digestive enzymes in pancreatic juice. Carbohydrate digesting enzyme, pancreatic amylase. It breaks down starch. Protein digesting enzymes, it is formed in the inactive forms. Trypsin from trypsinogen, chymotrypsin from chymotrypsinogen, carboxypeptidase from procarboxypeptidase, elastase from proelastase. Fat digesting enzyme, pancreatic lipase, it breaks down triglycerides. Nucleic acid digesting enzymes, ribonuclease, RNase, which can digest RNA, and deoxyribonuclease or DNase, it can digest DNA. Enzyme activation mechanism. Protein digesting enzymes are secreted in inactive forms to avoid self-digestion of pancreas. Trypsinogen, it is inactive and is activated by enterokinase, enzyme from small intestine brass border. It converts it into trypsin. Trypsin then activates chymotrypsinogen to chymotrypsin, procarboxypeptidase to carboxypeptidase, proelastase to elastase. Pancreas also produces trypsin inhibitor to block any trypsin that forms inside the pancreas by any mistake. This image shows the liver and 5 peritoneal folds or ligaments that support and connect to the diaphragm and abdominal wall. These ligaments include the coronary, right and left triangular, falciform and round ligaments. The liver is covered by serous membrane called the visceral peritoneum. Liver. It is located in the upper right abdomen and covered by visceral peritoneum and dense irregular connective tissue beneath the peritoneum. The lobes of the liver, it can be divided into two main lobes, right lobe which is larger and the left lobe is smaller. Falciform ligament, it separates the right and left lobes. It attaches liver to the diaphragm and anterior abdominal wall. Quadrate and caudate lobes, anatomically part of the left lobe based on the blood supply. Other ligaments, ligament teres, a round ligament, a remnant of the fetal umbilical vein found in the free margin of the falciform ligament. Coronary ligament is attached to liver to the diaphragm. Functions of the liver or bile secretion. Liver cells or hepatocytes secretes 800 to 1000 ml of bile daily. Bile is yellow, greenish or brown and has a pH of 7.6 to 8.6. The main components of bile are following: water, bile salts, cholesterol, a phospholipid named as lecithin, bile pigments especially bilirubin, and ions. Function of bile: it is excretory and digestive in function. Bilirubin is derived from the breakdown of heme from aged RBCs. Iron and globin are recycled. Bilirubin is excreted in bile, broken down in the intestine. A byproduct of starcobilin gives fecal matter or feces to brown color. Role of bile salts: it is made from cholic acid and chenodeoxycholic acid. It helps in emulsification. Basically, it breaks large fat globules into smaller ones. It increases surface area for pancreatic lipase to digest fats. It helps or assists in lipid absorption in the small intestine. Gallbladder structure: it is located beneath the liver. There are three main parts: fundus, broad end projects below the liver margin or body. It has central part. Neck, it has tapered end. It connects to the bile duct. Gallbladder function: it stores bile when digestion is not occurring. Sphincter of Oddi: it is located around hepatopancreatic ampulla and closes the direct bile into the gallbladder. It releases bile into duodenum when needed during digestion. Metabolic functions of the liver: carbohydrate metabolism: it maintains normal blood glucose levels. Low glucose, which converts glycogen into glucose, amino acid or lactic acid, and into glucose. High glucose, it converts glucose into glycogen or triglycerides for storage. Lipid metabolism: it stores triglycerides. It breaks down fatty acids to generate ATP, synthesizes lipoproteins or transport fats, cholesterol, bile salts from cholesterol. Protein metabolism: it deaminates amino acids. It removes amino group for energy use. It converts toxic ammonia into urea, which is excreted in urine. It produces plasma proteins like albumin, prothrombin, fibrinogen, alpha and beta globulins. Processing of drugs and hormones: it detoxifies alcohol and drugs like penicillin and erythromycin. It alters or excretes hormones like estrogen, aldosterone and thyroid hormones. Excretion of bilirubin: it takes in bilirubin from blood and secretes it into bile. Bilirubin is broken down in the small intestine and excreted in feces or fecal matters. Synthesis of bile salts: it is made from cholesterol. It is essential for digestion and absorption of fats in the small intestine. Storage function: it basically stores glycogen, vitamin A, D, E, K, B12, etc. and minerals like iron, copper, etc. Phagocytosis: it has Kupffer cells, special liver macrophages which remove old RBCs, WBCs, bacteria, etc. Activation of vitamin D: liver along with skin and kidneys helps convert inactive vitamin D into active form or calcitriol. Small intestine structure and function. Structure of small intestine: basically it is the main site for digestion and absorption of food or nutrients. Its length is around 3 meters in a living person. It has three regions like duodenum, which is the shortest part, about approximately 25 centimeter. It is a C-shaped structure. It begins at the pyloric sphincter of the stomach. Jejunum: it is approximately 1 meter long. It means empty, as found in cadavers. Ileum: it is the longest part, approximately 2 meter, ends at the ileocecal sphincter connecting to the large intestine. Intestinal juice and brass border enzymes. Daily secretion: 1 to 2 liters of clear yellow fluid. pH is slightly alkaline, pH is 7.6. It contains water and mucus for lubrication and absorption. 8. Brass border enzymes are present on microvilli of absorptive cells, allowing digestion on the surface of cells. Types of brass border enzymes: carbohydrate digesting enzyme like alpha dextrinase, maltase, sucrase, lactase. Protein digesting enzymes like peptidase, aminopeptidase and dipeptidase, nucleotide digesting enzymes like nucleosidases, phosphatases, etc. Mechanical digestion in the small intestine. Segmentations and migrating motility complex MMC, both of them plays a important role in mechanical digestion in small intestine. Mixing movements but not propulsive. It is the important phenomena of segmentation which occur in the distended areas which is filled with chyme. The food is mixed chyme with digestive juices and expose it to absorptive surfaces. Its speed in duodenum approximately 12 per minute and ileum is approximately 8 per minute. Migrating motility complex, a type of peristalsis that starts after most nutrients are absorbed. It pushes remaining chyme along the intestine. It takes 90 to 120 minutes to reach the end of the ileum. Chyme stays in the small intestine for approximately 3 to 5 hours. Chemical digestion in the small intestine. Digestion of carbohydrate begins in the mouth with salivary amylase, but this enzyme is inactivated by stomach acid HCl. But in small intestine, pancreatic amylase breaks down starch or glycogen, but not the cellulose. There is also brass border enzymes like alpha dextrinase which breaks alpha dextrins to glucose, sucrase breaks sucrose into glucose and fructose. There is lactase which acts on lactase which acts on lactose to form glucose and galactose. There is maltase which acts on maltose or maltotriose and converts into glucose. End product are the monosaccharides like glucose, fructose and galactose. Digestion of proteins, which begins in the stomach with pepsin, proteins converted into peptide and continues in small intestine with pancreatic enzymes and brass border enzymes. Pancreatic enzymes are trypsin, chymotrypsin, elastase, which breaks peptides between specific amino acids. Carboxypeptidase, it removes amino acids from the carboxyl end. Brass border enzymes, aminopeptidase, which removes amino acid from the amino end. Dipeptidase, it breaks dipeptides, free amino acids. End product, single amino acid. They can after digestion, single amino acids are formed after digestion of proteins. Digestion of lipids. Main dietary lipids are triglycerides. It begins slightly in stomach with lingual and gastric lipase. Measured digestion in small intestine by pancreatic lipase. Bile salts emulsify large lipid globules into smaller ones. Emulsification. Bile salts have both hydrophobic and hydrophilic parts. This increases surface area for lipase action. End product, fatty acids and monoglycerides. Digestion of nucleic acids. Pancreatic nucleases. Ribonuclease, it digests RNA. Deoxyribonuclease, it digests DNA. Brass border enzymes, it has nucleosidase and phosphatase which breaks nucleotides into pentose sugar, phosphate group and nitrogenous bases. The end products after the digestion of nucleic acids are absorbed via active transport. So I hope this class helped you clearly understand how digestion continues beyond the stomach, how the pancreas, liver, gallbladder and small intestine work together as a powerful team to complete chemical digestion and begin food absorption. If you enjoyed this simplified step-by-step breakdown of how your body processes food in the small intestine, don't forget to like, share and subscribe for more such student-friendly lessons. Stay curious, stay consistent, and I will see you in the next class. Thank you.